So our next guest is Hunter N. Hines, who's a PhD from the Harbor Branch Oceanographic Institute, Florida Atlantic University. But you also run the Instagram account at Microbial Ecology, which is another reason why you're here. And yeah, thank you for joining us in the Microscope Hour. In a second or two, I will enter into the Zoom call on my phone. I have a microscope here, which you might be able to see. If I do this, maybe you can just about see. Oh, that's Eleni and my microphone. Here we have a small, but functional microscope, which we can use. And yeah, we can kind of see and delve into a bit more detail about how to use microscopes to communicate microbiology. Yeah, so I have a few questions for you.
All right, I'm here.
And the first one is, so you now have this Instagram account, which, in case people haven't seen it, takes like short and interesting videos of microbes living in pond water and samples you find, and you post it as an Instagram, and you've amassed a huge number of followers. But what got you started doing this? Why did you think, oh, I know, I'll film these things and I'll put them on Instagram?
Right. Well, first of all, hopefully all your followers right now are following me at Microbial Ecology. They're on their phones, busy.
Yeah, find it.
They've done that, and they can pay attention to my explanation. So basically, I started this during my PhD. So back in 2017, I started the account. Yeah, I was generating a lot of images for the project, and I was really kind of interested in what I was photographing, and I thought that I should maybe record some of these for myself just to look at later, to use for lectures. And I thought, well, why not just try to share these? And Instagram was a really obvious way to do that. Just super easy, a couple clicks, you can share it directly from the microscope. And I think that's why I got such an interesting, and a large number of followers, because not many people at the time were doing that. And it's kind of this, unseen world. And I had access to such amazing, research grade equipment. Why not share it? So luckily it took off.
Yeah, and it's, probably brought microbiology and microbes to like so many more people than almost any other method I've seen.
Yeah, hopefully.
Because you've got now nearly 140,000 followers.
Yes.
So within that, who do you think what's your main audience? Like, who are these people, and is there any general trends, or is it just a mixture?
Yeah, well, Instagram gives you some analytics. So the majority of my followers are between 18 and 34 years old. At least that's how they identify. I'm sure you can type in anything you want there. So yeah, it's younger people. That's who's on the app primarily. But interestingly, and I've read about this in a couple of papers, a lot of it's kind of a worldwide amount. It's not just 100% Americans, it's all over the planet, and that's what's kind of exciting. It's people in areas which may not have readily available access to this research grade equipment or stuff like that. So it's great as long as they have internet access, they can just click and take a look down a $50,000 microscope essentially.
Yeah.
So
I think that's one of the most amazing things is, like, if you are a microbiologist or you're working close to microbiology, you get so used to just looking at things down the microscope and being quite blasé about this massively amazing world that exists down there. But if most people have no connection to a powerful microscope, so when they see these things, they honestly are surprised and stunned by what they see, aren't they?
Yeah, and that was something I noticed, giving just in-person tours of the lab for different donors or prospective graduate students. They always had this kind of this awe and this amazement when they looked down the microscope. So I thought, how can I extend that beyond just 5 to 20 people a month? And social media is a great way to do that. You never know how many people are going to watch, what might go viral, but why not put that out there for anybody to see and incorporate into their lives?
So, like, what happened when you started this? Did it quite quickly become big, or were you, low key for a while, and did it all suddenly change in a year? Like, what was the kind of...
Honestly, it took me a little while to figure out how to really use the app properly. Once I optimized that, it probably got to 10,000 within a few months, which was kind of slow. And then once you hit that point, it just kind of went a lot more viral. The different larger entities can pick up your profile, and for them, a lot of the online media, it's very easy. Just here's a couple links, here's an interesting microbe, boom, 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 and that shows their whole audience. So that's how it kind of got a lot more viral, and that's exciting too, because these are people who for sure wouldn't maybe Google ciliate symbiosis. They're not looking for this, but it's kind of thrust upon them and they still find it interesting. So that's a great way to kind of bring in people who aren't necessarily going to sign up for a microbiology conference, but they are interested in the natural world.
Okay. So then, I mean, you said that you've now honed your presentation and stuff a bit over these years compared to your first effort. So what advice would you give to any scientist who, they might have some microscopes, they might want to start using it to communicate? What are the main things they should consider when they're starting this?
Yeah, well, Instagram is so visual, isn't it? I think the most important thing is just getting a really nice looking image. So that really goes with having a nice adapter for the microscope and using, the best images you can. Obviously, something really clear and bright is what everybody wants to see. And for me, I've had the most success just being very brief in my captions. I think people have enough having to slog through textbooks in Bio 1. They're done with that. They don't care about it. But, seeing a Paramecium as a living organism is really interesting to them. Perhaps they've only looked at a diagram previously. So it's kind of bringing that to life. And in my posts, I make sure to, not only keep it brief, but just highlight a couple of the key factors. And then if somebody's interested in that, they can then Google those further. So I'm not giving them a Wikipedia page. It's just, here's the creature, here's something to look at, do your own work essentially. And I think that's kind of a great way to learn. It's self-motivated.
Okay. So I just wanted to know, do you have any of your videos preloaded, perhaps, that we could have a look at, just to give someone an idea of what kind of, because it's, these are all very bright, colorful, well-framed images. If you can't see the Instagram right now, no worries. And so there's a certain approach that works well, because you have to make sure that you're seeing stuff in a nicely focused, clear, and sort of detailed way.
Right. So I'm trying to see how I have a few of the videos on my desktop, how I can upload them to you.
So virtual background's pretty good, because then you're in the video.
Yeah, it wouldn't actually work with my computer wouldn't work on my machine. It's a MacBook.
Well, maybe you can share a screen, and then actually that will probably be able to show at least some of the images.
All right, it says host disabled attendee screen sharing.
Oh, okay. Yeah, we've been in a new call, so there we go. That should now allow you to do it. Yeah, we've been live streaming for 7-hour chunks, so we've just begun a new one. So it's all back to default settings a bit.
Right. All right. How does that look?
Yeah, we can see your desktop. Some beautiful underwater scenes.
Oh, yes. That's the Florida snorkeling view. All right, here we go. Here's a video. Is that playing for you yet?
Yeah, it is. So we've got some kind of ciliate.
Yeah, this is a ciliate blepharisma, so it's naturally pink. This is in a really kind of bright phase image. You can tell it's phased because the bacteria in the background are showing up as really nice and black. And to the upper left is kind of a worm which has got itself kind of stuck in the air bubble on the slide, and it's kind of wiggling around. I like this image quite a bit because the top part of the ciliate, you can see really obviously these hair-like projections, which is what makes it a ciliate. That's how they move with cilia. So yeah, it's a super common, lab rat organism, but I think it's nice just to show it, moving and interacting with a multicellular organism, which is, this ciliate's actually much bigger despite being a single cell, which is kind of fun.
Yeah, that's something we looked at earlier today. We were looking at different scales, and there's amoeba the size of dust mites, even though one is single celled and one's multiple cells. They're the same size.
Some of the ciliates get to be well over 1-2 millimeters in size, so you can actually see them with the naked eye, which is really cool. This is always popular. This is a tardigrade, obviously. So this is a water bear. It's multicellular eukaryote. It's an animal. You can see the head is towards the bottom there. It has 8 legs, claws. You can see the cyanobacteria. It's ingested in the center, that kind of yellow mass. All the little circles are storage cells, which are kind of fun. And you can see the 2 little black dots. Those are the eyes, and those eyes are each a single cell, which is kind of interesting. These are these, indestructible creatures, which of course are quite fragile, but they are resilient to a lot of things. They can survive ionizing radiation in space, all that kind of thing. This is just found in a freshwater spring here in Florida on the microbial mats. They're found everywhere, but they're not as easy to find as people hope they are. But there's a few really great sites here in Florida. Tardigrades are always popular, I think, because they have a really interesting, upsurgence in media these days. So people are kind of familiar with the term and they want to actually see them alive, not just a TEM or an SEM image where it's just frozen in black and white. To see it crawling is kind of fun. I think.
So you've actually, in the past, you shared one of these videos with me for a video I made on a tardigrade, a little piece of footage. And we actually, when we were testing all this live stream software, looped it for 24 hours on our channel, just to kind of have some video playing. And
cool.
yeah, 888 people watched this looped tardigrade wiggling around. And actually someone sent me an email being like, is this meant to be on the channel? I'm so confused. And I was like, no, don't worry, it's a test.
Yeah, I think tardigrades should be used more often for 24-hour loops. I mean, why not, you know?
Yeah.
Let's see, play this one. This is a kind of fun ciliate I just took out of a saltwater aquarium. You can see how kind of, you know, plastic these things are. It left behind a bit of algae it ingested, it didn't need anymore. And this is another phase image. Just see the amazing detail of the cilia around the mouth. And these really are complex creatures despite being just a single cell. You can see the huge mouth at the top of the screen there.
Yeah, it's huge.
Yeah. And you can see all the, you know, these are just natural color. It's just brought to life with the polarized light. So these are all different types of, you know, protists this thing is ingested. So you can see a diatom in there, various species of algae, all that kind of stuff. So it's really nice kind of looking at this. These are the foundation of food webs, obviously. So to see these interactions at this level is quite interesting. This being obviously a predator of algae, and then larger things would eat this cell. So I always find that fascinating.
Are all of these from samples that you have found in Florida, or do you ever get someone to send you something from further afield?
Yeah, you know, it's a little bit tricky receiving international samples to a laboratory university in America. A lot of awkward rules with that. So most of the stuff I post are things that I've personally collected here in Florida. And obviously for my PhD, I was looking at ciliates in freshwater and terrestrial systems. So I did a lot of sampling, took a lot of images, and that's where all this content came from.
Yeah, very cool. Okay, one of the things I'm going to do now is I'm going to join this call again on my mobile phone, and then we're going to try and see if we can do some live microscopy. Ooh. One of the things I need to do is mute the volume. There we go. And I also need to start video. So there we go. You can see me a bit blurry from beneath. Yeah. Beautiful angle, but we need to flip this round. Cool, okay. So this is the setup we are in today. We're in the studio room with many screens. And then we've got Eleni, our science communication assistant here, who's doing some backstage stuff. And then here's our...
The thing kind of froze. Hopefully you can still see me though.
Yeah, just about.
I can hear you, but yeah, it's frozen.
Ooh, okay. It's having to adapt to many streams. I'll try and just stabilize this in the adapter then.
Are you still there, Hunter? Ah, now you're moving.
You guys still there? Can you still hear me?
Yeah, I can hear you now. Can you hear and see us okay?
Yeah, I can see you. Yeah, it just kind of froze a little bit.
Cool. I think it was just adjusting to...
yeah, it's Zoom at 10 a.m. in America, so everybody's logging on right now. It's never stable.
Yeah.
Well, it seems like a good time. It's actually sometimes not a good time. Right, okay.
Yeah, it's incredible, the images, Hunter. Like you're saying, you get a lot of interest from non-microbiologist people who don't understand it, but I was doing bacteriology research, and this is something that I never saw in my research at all. And it's, you forget sometimes that these things exist and how fascinating.
Yeah, and I think it's nice, just the more microbiology you watch as a professional, the more you kind of learn and incorporate into your own studies. You know, I got an email from somebody who was a professor in UK, and he said, man, I didn't know these ciliates ate diatoms. I said, well, what do you mean? Of course they do. He said, well, I just never thought of it. And so I had posted a video, and it's full of these diatoms, which is what he was working on. So it was kind of interesting little just collaboration there, just to kind of expand his realm of thinking. This is a PhD scientist, so I think it's great.
Mm. Okay, I'm going to just, spotlight, I'm afraid, the microscope video. So we're going to just appear above the top. If I just check what the live stream looks like from anyone else's perspective. Yeah, okay, so we'll appear when we're speaking in the top right-hand corner, or maybe they'll just hear us. I'm not sure. Let's have a look.
I'm sure they're more interested in seeing the microbes than me.
I think so, or than myself. Right, let's do a test because we have some just sort of like less animated slides that are just, things secured here. So let's start off with something historical. So back when, the first microscopists were looking at stuff, we obviously had Micrographia by Robert Hooke, and one of the famous illustrations was that of a flea.
There it is.
So here we have a flea. How's that coming through? Yeah, okay, cool.
Brilliant.
So I'm on, I think, the lowest setting.
Yeah.
So it gets a bit dependent on how much you can fit in between the slide and the microscope, but we can zoom in a bit further and see some more detail. Cool, okay, so here we have some flea legs. You can see some nice hairs.
Yeah, beautiful image.
Yeah, so I gave the microscope a big clean using one of these dust cleaners before today, and it did really help.
Are they sponsoring the video?
Sadly not.
No, pity.
Okay, so that works well. Another cool thing we can look at, I've got a few set slides, and I do have some live samples we can look at as well.
All right.
And then we can use your expertise, Hunter, in trying to guess what we see on camera. Although it won't quite be as clear as what you've produced because it's not a £50,000 microscope, sadly. Here we have a mosquito on the way. So yeah, there we go. Here's the tail.
Nice.
And the body of the mosquito. And you can see kind of the digestive tract,
Sure.
which I guess is probably full of blood when it was fixed because it's very dark. And you can see some eyes. One of the fun things as well, although this isn't very scientific, is I can put colored filters on to try and make it look a bit cooler.
Oh, nice. I think it's worth noting too how, you're a professional, but how easy it is just to kind of get a live image from a microscope. So I think a lot of researchers, especially students, should maybe think about just how easy it might be to share their work.
Yeah, especially nowadays. You just need a smartphone and Zoom.
Yeah,
and the Wi-Fi, and you're there.
Yeah, I mean, Twitter, Instagram, it's all super easy. So why not post what you're doing? Otherwise it just remains private. You do all these years of work, and who's really going to read your dissertation or your papers? Not many people. But social media, you can bring in everybody, which is kind of fun.
Well, this is something I think about in communication. It's like you have to think about the cone of communication. There's like the initial field who might be 5 people who will read your research, but is there any reason to favor them beyond the rest of anyone else you could reach? If the research becomes more communicable, you get a huge number of people then understanding what you're researching.
Yeah,
okay,
I think, recently I posted a bunch of silly videos, which were from a chapter of my PhD, and they were, I did publish a paper on it recently, and I shared that paper, but I think people are a lot more interested in just watching the thing living. If they want to go look at the genomics of it and all that stuff, they can read that paper. They can read my methods if they really want to, but they just want to see the creature and see it for something unique. And I think that's what social media brings to the table.
So here we have coming up next, there we go, an ant, which I really enjoy this slide.
Oh yeah, nice.
Yeah. So one of the things that I guess you might not know if you haven't used a microscope is you're kind of also focusing at a point within the slide. So you can kind of move up and down through the animal by focusing on the top and then focusing through the animal, and you kind of like move down in layers, which is kind of strange to think about, but cool.
Yeah, a lot of people, they ask me in comments or messages, why is this 2D? And I said, well, no, the microbe's not in 2D. It's just, either very thin. Well, that's kind of, think about how a camera works. I look like I might be in 2D, but actually I am in 3D. It's just the way these images work. So the camera of a microscope is similar. You can go up and down through the focus, and that's what people kind of lose perspective on. But interesting point to make, yeah.
Yeah. So I have also got some live samples, but I'll just do one more of these fixed ones just to see, because we also have, these were insects, but it can also be quite good to show plant life.
Yeah.
And then we can see kind of the inside of a cotton stem somewhere over here. Which way am I going? Am I in the right direction? I think I would be told off for not keeping my slides very clean if this was a professional lab. Here we go.
Oh yeah, nice.
Yeah, so here you can see kind of the cells inside a plant. And actually they've stained some of them. So I think you've got kind of the xylem are stained, maybe a bit darker.
Right. Yeah, pretty.
Yeah.
Very nice.
Cool. Okay. Now, the next one is kind of a collection of different weird things that I put together in this little box. Although one of the things that happened was, if you can kind of see it here, I had a beautiful collection of salt crystals, dried moss. And then last night I put some pond water in this one. What happened is all the moisture kind of evaporated, moved down the chamber with the lid on, and then it's dissolved the salt crystals and rehydrated my moss, which isn't ideal, but we'll have a go. Okay. So there are a few mosquitoes down here, which are real mosquitoes in the sense that they are from my flat from this summer.
Oh, and, nice.
Yeah. So you can also illuminate from above.
Oh, yeah.
Yeah. So now you really get a sense of the kind of 3 dimensions, I guess, because you can kind of move down the animal.
Yeah, and how terrifying mosquitoes really are. I mean,
yeah.
Oh, my screen's just gone dead. Just getting up. Here we go. So yeah, I mean, obviously your research has helped your Instagram page, but has there been any reverse relationship? Has running this account helped your own research in some ways?
Yeah, I mean, just, self-servingly, it was, a chapter section in my PhD. So that was nice, kind of talking about social media outreach. But I think just overall as a scientist, it helped me just kind of understanding the relationships between these different trophic levels and almost just identifying what I was looking at. I mean, I was very specialized in these freshwater ciliates, but finding out what else was in the water, what else in the community, and how they kind of interrelated. To explain that in the posts, I think was something I would not have done had I not been posting, daily on social media. So I think overall it kind of rounded me out better as a microbial ecologist.
Oh, that's good. So then, as a microbial ecologist, if there's one thing people should know about microbial ecology, what do you think it should be?
Oh, I think it's that microbes are everywhere and they're incredibly important for the survival of life on this planet. Microbes tend to get kind of a bad rap. They're kind of like sharks here in Florida. You only hear about them when they hurt or kill somebody, but really they're here all the time and very important in ecosystems and within us, microbes. So I think it's good to remember these things are a healthy part of environments and they're incredible.
They definitely are. Right, okay. I think we've seen now enough of this kind of thing. I think it's time to try and find some microbes.
Ooh.
So this might take a second or two because I have to prepare a slide. Yeah. But do you
we'll judge your slide preparation skills then live?
Don't. A long time.
No pressure. How did you get into specifically this area of research, Hunter?
Oh, so I was actually doing a master's degree at Bournemouth University in England, and my supervisor there was Dr. Genevieve Esteban, who's kind of a world expert in freshwater ciliates. So I just did my project with her and just really kind of fell in love with the microbial world, and that continued on to the PhD. And I've been looking at microbes ever since, since 2013, I guess. Yeah.
I'm just going to show people roughly how you set a slide. And you can judge me as well. So I've made it the spot.
Always.
Yeah. So we have, well, I'll just show people what we've got here. So this is a sort of collection of gloopy pond water that's
nice
been
color.
kind of, sat on my desk in anticipation of today for probably 3 weeks, 4 weeks. So a lot of multiplication of microbial life has gone on in here.
Nice.
And we're going to try and see if we get on a slide. So one of the other things I have is a little pokey stick, which is very helpful. So we're going to take a drop. Shall I give it a stir?
Is that a weld slide or a regular, just flat slide?
So this is a flat slide, and then I've got a little glass cover slip like this. Which will go. I mean, it would be nice if I had little divots for a bit more of a to capture, because this one does have the tendency to overflow. But if we put a drop like that, and then, the trick is to not get air bubbles and not get fingerprints. I'm failing at both. Okay. Right. So now we have, hopefully, some microbes trapped beneath the pieces of glass here.
Nice flat mount.
Right. now switch back to the microscope.
Maybe we should call this video Hunter Hines Reacts to Microscopy Setup.
Yeah. Yeah. It's a shame you can't join.
Definitely overflowed the slide. Let's not mess around.
Yeah, it's a shame that you can't access your own microscope on this auspicious day.
Yeah, sadly, no. My lab is fairly shut down now. Coronavirus is fairly serious here in Florida, so we're minimizing everything.
Yeah. I never realized, until I had a little look, how many people live in Florida. It's just huge. So many Americans, like a fifth or something, live in Florida. It's ridiculous.
It's a big state, yeah.
Yeah. Right, okay. So I have got a bubble under the slide, so that's already lost marks.
Oh man, no for two. Yeah.
Yeah. We do have some debris, although it's not quite as thick and rich with microbial life as at the time I did it yesterday. So maybe I'll try and doing this again, but let's have a bit of a search because we might find something cool. I think I need a bit of a gloopier drop of water. Do you know what I mean? I think I was taking it from a bit too near the top. So I'm going to set this one again because we need some more greenness.
Yeah, try to get maybe the very bottom, the bottom layer. See what's
Yeah.
down there.
I think if we get...
This is kind of the fun thing of, sampling for microbes. It's almost like fishing. You ever go to the ocean and you never catch a damn thing. There's fish there. Very similar for microbes. They're all over in this system, but whether or not you're going to get the targeted microbe is always the question,
Yeah.
which we're demonstrating here.
Okay, so you must spend quite a lot of your time out there, Hunter, like looking for kind of sampling and doing field work and stuff like that.
Yeah, a lot of my time, and I had 4 major sites for the PhD, which I sampled a couple times weekly. So there's a lot of water samples, recording presence, absence, all that kind of thing, describing new species. It's good fun, but a lot of time on the scope. Yeah.
Oh, there's something. There's
There you
something
go.
alive.
Something alive.
Let's go in one more level. See if we can find something. Something is moving there, isn't it?
Yeah, there it is.
Yeah,
It looks
I think
like a
it's
little rotifer, I would say. Yeah.
yeah, I think you can also see it filtering the food.
Yeah, so it has that pinwheel-like mouth, which is ciliated, and it brings in the current, and it can also use that to really jet across the slide, as well as its little hook foot.
Ooh.
That might
So one
be
of the
a little-
problems here is
yeah, I
that
think that's too close. You've hit the
it's
slide.
too close, yeah.
Yeah.
And it's now formed a drop on my microscope's
That
lens.
was so bad it's froze the screen now. Hopefully you can still hear me.
Yeah, you can still hear you.
So a murky amount of dishwater. Okay, let's go back to the previous one. There's a lot of movement now within the water, but we have now got, a denser mat of microbial life. So there's some other things in here. So there's some more filamentous stuff.
Oh yeah.
Yeah. What do you think those are going to be?
Yeah, probably some type of cyanobacteria, the filaments. It also could be maybe like a Vegetoa, a filamentous bacterium. A little hard to tell from this image, but I would go with cyanobacteria based on the coloration of the water.
Yeah.
As long as you say it confidently, people will trust you, I think, as well.
Yeah,
So.
that's true, isn't it?
Yeah.
What I like is that because there's so much debris, you can see the flow of water into the Rotifer's mouth.
Yeah, exactly.
And you can kind of see the flow patterns, which is kind of cool.
Yeah.
Right,
This is coming from your backyard pond, or where was this sample from?
So this sample is actually, this is kind of weird, grown from like some disgusting murky plant water that was sitting on a windowsill.
Oh,
And it kept it in this jar for some reason. And it wasn't very full of anything particular, but there must have been a few things in there, and now it's full of life.
yeah. Yeah, rotifers are pretty hardy. I mean, they'll take over a lot of abandoned cultures. Certainly in my lab, you'll have them full of rotifers eating all the bacteria matter, all the algae.
I mean, I haven't ever found a tardigrade in any samples I've been looking at yet. So do you have any advice for where we should go for them?
Yeah, I mean, they say you should look in moss, and then just put a little bit of water in there, and then tilt the dish, and then just take the water out, and hopefully the tardigrades will be in there. for my own work here in Florida, I have several ponds I sampled for years and never once found a tardigrade in. And a couple of springs I go to, and every single time it's just full. You know, there's hundreds of tardigrades per milliliter essentially. So it just depends on the habitat, really. Even though they can thrive everywhere, it's kind of like the fishing analogy. You just never know if you're going to be able to catch one.
Yeah.
But yeah, I would try. If you're somewhere are you calling from somewhere in Northern Europe? Are you in UK? You're in Netherlands?
So I'm in the Netherlands and Ben is in Sweden.
Oh, okay.
Yeah.
So yeah, I would definitely try some woodland moss then and see what's in there. Yeah.
Because they are in every continent, right? Tardigrades. So
yeah,
wherever you are, you can have a go at finding them.
Never far from a tardigrade. Yeah.
Yeah. It's like a much nicer version of a mouse or a rat, really.
Exactly. Yes.
Okay, so here we have some more spherical-like organisms, which are also moving.
So I would say those are probably some type of little ciliate, maybe a flagellate. I can't really see too clearly, but
yeah, it's not
yeah,
quite
the one in the middle is probably some kind of a little ciliate cell. Yeah. And then
So
there's the single-celled algae. I would see the different circles there, which aren't moving. Ooh. Big rotifer.
Look quite monstrous, don't
Yes.
they, when you get up close?
It's like a whale, yeah.
Yeah. Brilliant.
So we've had some cyanobacteria, we think some ciliates, definitely some rotifers.
Yeah. So when I did my project, I only worked on the really large ciliates, these flagship species, because there's so many tiny ones. They're very hard to identify, hard to manipulate. I looked at the really large ones, so it'd be kind of fun if we find one of those, but nice rotifer, yeah.
One of the problems is that this magnification, the lens is so long, it can bump very easily into the slide.
Especially when it's overflowing.
Yeah.
The shade.
I'm very sorry, guys. But yeah, here we have some, I don't know, they're kind of paired into little groupings. Do you see?
Oh, yeah.
Yeah. It could be colonial algae. It's a little. It's a little blurry on my screen. That's my defense. I get this totally wrong. If some expert is looking at their screen and saying, "What the heck is he talking about?" But yeah, very nice.
But you can kind of see how everything moves if I tried to zoom too much in a certain direction.
Yeah.
Just applying pressure. Right, I'm going to move this back to another microscope. Standard. And we can come back to this one, but there are some other things we can look at. And one of them is I have been growing this bizarre biofilm sludge.
All right.
There's been some peppercorns in here, like, just black peppercorn. And it's been sitting in this jar for, again, a couple of months. So we'll see what these kind of, gloopy, biofilmy things look like. But I'll try and do a smaller drop this time.
All right. Describe the fragrance when you open it.
Yeah. Let's have a smell. I'm the only one who can experience that, so I'm the only one who has to suffer it. But we'll have a go.
Yeah. Well, miles away.
Right. Very professional saliva clean, and let's have a go. Where's my other pet gone? Somewhere. Here it is.
Can you culture any of these larger ciliates, Hunter? Can you actually have cultures going in your lab, or do you have to sample them every time?
Yeah, I did culture a few of them. There's some old methods which still work. The only thing is for culturing, especially these large cells, is you have to kind of pick them individually, and you get some contamination if you're doing an environmental sample, and then something else will bloom, like a rotifer, and then you have a pure tube of rotifers, which you're not trying to work on.
Right.
So I have given it a smell, and it just smells like pepper, like fresh pepper. Nice. So that's not too bad. But if we can have a look, you can kind of see in here. I don't want to pour it on my computer. Right. There's lots of film that's formed.
Oh, yeah.
A
lot of life in there. Okay. Let's take a look.
Trying to get some of the thicker sediment because that's the more interesting.
Yeah. to your other point, there are some microbes which I just cannot culture, and that makes them a lot more difficult to work on. You're trying to get their molecular sequences, you're trying to really image them. You maybe only find 1 or 2 per 500, milliliter bottles, and so those are very difficult to work on. And then sometimes, there was one I was hunting for a long time. I saw it once, then I didn't see it till the following year, and then I had a bottle just full of them. I mean, you couldn't look through the bottle; that was so dense with this particular cell. So you just never know. This is kind of the fun.
Cool.
Video. Okay, so this one, I've put less liquid on the slide, so hopefully we'll get a better picture.
All right.
We'll start maximum out, zoomed out. Okay.
Okay.
So we have some weird gloopy mats. I'm not sure if I find anything as big as a Rotifer because it's been fairly anaerobic in there. There's not been much oxygen for a long time.
Yeah, it might be just a bunch of, bacterial slimes, something like that.
Yeah.
Microbial mat. Yeah. Yeah, it looks like a really dense accumulation of bacteria, which are just breaking down those peppercorns in there.
Yeah, because there's different sugars in the peppercorns, which some favor and some do not.
Yeah.
One more. Can we go in one more? Maybe just about, because what's annoying is we can kind of see that at this resolution, this zoom, we're actually getting such a small amount of light, it becomes quite dim. Right. But we can just about see some things. What is quite fun is if we use different light filters, it kind of shows you different things.
Very nice.
Because it picks up different, blue's a bit confusing. Let's try. Pink is quite helpful. So yeah, this is kind of pushing the resolution of this microscope because these, I think you're right, are individual bacteria and we can just about see them, but it's quite hard to get much detail on them.
But quite cool. You can see how tiny these cells are, and then you look at the jar, it's just this kind of milky substance. So kind of amazing. Yeah. So many cells, you can see it so obviously.
We can also see if we, purely light from beneath. Oh, yeah, it is just a massive cells, isn't it?
Yeah, very nice.
There's a bit of green in here.
Yeah.
There's some darker objects as well, so there's some weird stuff in there. But do you have any idea what kind of bacteria?
Zero idea.
Zero idea. Because I mean, is this going to kill me if I get too close, or is it something that's perhaps fairly...
One way to find out, I guess. Yeah. How much longer do you have on the live stream? We'll see maybe in real time if you're going to...
Yeah, if I make it to the end, is a question.
Right. That's great TV. Who would turn away from that? Even poison at 10 a.m.
Yeah, let's eat random microbes. At least it proves our faith in microscopy and in microbes.
Right, next year.
I'm just cleaning up the slide that has the pond water on, because I think there's more moving things in there than on this one.
All right, let's look.
Yeah.
Yeah, there's just so many different species and so many phyla. You never know what you're going to run into, which is the kind of exciting part. Especially with bacteria, so many of these things look similar. You really these tiny little cells.
So other than tardigrades, have you found there's any life forms which people respond to best on your channel?
Yeah, I think it's based on kind of interesting coloration is always popular. Something's bright blue or bright pink, or different, forms of motion. Some of these big flatworms, they move such interesting ways. They're so kind of elegant in the water. Those always draw people's attention. It's something they haven't really seen before. That's a nice, really good image there, showing all the water currents being created by that rotifer.
Yeah, it is good, isn't it? Let's see if we can get in one more level.
Yeah, really nice.
I think I've just ruined it, I'm afraid. Sorry, guys. I was going for too zoomed in. Here we go.
Yeah, happens. Yeah, I think that lens is probably too close to the glass. It must be hitting it.
Yeah, it's a shame because we were getting some good shots yesterday. I'm not quite sure how it managed to focus, but we'll go back one level.
It's crazy.
Yeah, everything's a bit suspended in some flow.
It's fascinating to think. I mean, so I'm really inexperienced with these kinds of stuff. I was working on bacteria, but these rotifers are such, just like hungry little beasts. You just kind of see them creating the air current and filtering. And the way you're talking, Hunter, it's like, yeah, these are like the contaminants. This is not even the interesting stuff for what I do. Like the world, of kind of ciliates and, in these kind of samples is incredibly diverse.
Yeah, there's a huge diversity, and rotifers are kind of weedy species. They're able to move through the air. They can form these kind of resisting stages. So if you have just a backyard bucket, within a week you're going to have rotifers in there, pretty much no matter where you are in the world. And so that's kind of interesting. These things are always common, especially in natural environments too. So they're readily, they reproduce, they survive really well. So yeah, they're kind of annoying for me because they'll either eat or they'll just mechanically break into the ciliates I'm working on. So rotifers and I don't get along so well, but they're amazing to look at. Yeah.
You must be like, not another one. Because,
yeah.
I mean, so what's a lot
of pure cultures of rotifers.
Yeah. So what are the more rare kinds of microbes that you're hoping to find then when you're looking for something to post on the channel or even as part of your research?
Yeah, I mean, for my research, I'm always interested in these large cells, these really big ones, up to 1 millimeter in size, which is so cool for these single cell creatures. For the PhD, we discovered 5 species new to science. That was really fun because they were at that millimeter or larger size point. So I wasn't looking at these little 5, 10 micron cells nobody can even see. We were finding really obvious things which are very hard to miss in a sample. So that was cool to document those as new species because it was clear that nobody had seen them before, despite them being so common here in Florida. So that was nice for me just to see how kind of open the microbial world is and how much discovery there is to be made at these really obvious cell levels. So you imagine the little tinier zone, how much more discoveries there are to be made here by anybody with a microscope.
This is probably a stupid question, but when you do a sample like this, how do you know you've got a new species? What's the first moment you kind of think this has never been described before?
Yeah, well, luckily I had immediate access to my supervisor, Genevieve. Even though she was in England, I would just WhatsApp her a video. And so if she hadn't seen something, that was always a really good sign. But when we're looking at these cells, which are so large, there's very few of them, especially in the given genus. So you can almost rule out right away. For instance, we found one which was about 1.5 millimeters in size, and everything else in that genus was maybe 200 or 300 microns. So this thing is over 10 times the size of the known species. So that was almost right away. This is something new. So that was really cool. You can just tell right away. Yeah, and there must be tons of them out there, really. I mean, I just was looking at freshwater, warm ponds here in Florida. The diversity of the planet is such that there must be an incredible amount of species out there waiting to be discovered.
In fact, there was a talk earlier this morning which still fascinates me, and they were looking at yeah, you back there? We froze for a second.
Right. You got me back on. Sorry about that. You guys froze.
No worries. So did everything. Yeah, I'm just checking. I've got my Ethernet cable in. I think
Oh yeah.
Yeah.
Anyway.
Cool. Did you hear?
Sorry, guys. I must have missed the question. Go ahead.
No worries. I think yeah. I mean, this morning we had a talk about multicellular magnetotactic bacteria, which blew my mind. And I just never heard of such a concept. So that was pretty fundamentally amazing.
Yeah.
And they were new species, yeah, or at least relatively new.
Yeah.
So in your process of filming, have you developed any special techniques for kind of making stuff look good? Are you just very adept now at using the different filters and so on?
Yeah, it's kind of a bit like driving. The more you do it, the better you get. A lot of my stuff on Instagram is DIC filter. So it's that differential interface contrast. And that really kind of brings out the 3D, it makes it really pop a little bit. It also takes out some of the background noise. So you get almost see the gray to blue scale. And it really shows the detail, especially for these single cells. And I think that's really fun. And yeah, just showing these things, here's a rotifer moving just as living, not just here's a boring black and white, drawing in a textbook you have to memorize. I think it's a lot more meaningful for people to see these things living, whether or not they've heard of them or not.
Absolutely. So here we go, we've got a nice rotifer stuck in between some cyanobacteria. So I'm guessing it's trying to eat loose bacteria or something within the cell.
Yeah, it'll pull in any kind of particles that, like, bacteria, different algae cells, all that kind of stuff. It'll eat ciliates as well. Yeah, a bunch of them in there. Nice.
Yeah, let's get this focused. I quite like this view because it gives the kind of impression that you're. Let's try it. Which one of these is best? Yeah, that you're looking at. It's no longer a cross section. It looks a bit more real under this.
Yeah. Definitely. And that kind of view, that imaging is nice if you're picking out cells which have different colors in them. Maybe you can just look at for, the gold or the blue. And if you're picking out individual cells for research, this kind of black scale is very helpful.
Because we also have here, I think these are groups of algae, kind of bigger spherical cells. Can you see my mouse? Or is it? Yeah, no, maybe not. But and then we've got the cyanobacteria wrapping around it. So this has just been growing on my desk. I'm not quite sure what to do with it now, but it is fairly good.
Yeah, I think keep it going, right? Why not?
Yeah, I mean, I think for me, the holy grail is if we find some tardigrades and start videoing those.
Yeah.
Because that would be fun. Because actually, do I have it on this computer?
Moths and take a look. Yeah.
Because there was, actually, I think I might. I'm just going to see if I can find it. There was so we did this small series called 52 Microorganisms where we made a video a week about a new microorganism. And I think you just published a paper in FEM's Microbiology Letters about using Instagram for...
last year,
Yeah, and at some point I think I was in contact with you and you lent me a piece of tardigrade footage which made it into the video.
I did, free of charge.
Yeah, you didn't at all. Very generous.
No royalties or anything.
Yeah, okay, so if I now...
Oh, nice. Yeah, so there's the electron microscopy image of a tardigrade, which people have seen, and then hopefully the video will bring it to life. There you go.
Wow, here we go. So I'm now amongst tardigrade. So this is the one we had on loop, but you can kind of see all the cells inside his body, which I find very strange. And then you got the mouthpiece.
Yeah, the mouth, a little brain up there, yeah.
And this is his belly. Yeah, and this stuff here is it's food. Hang on, whatever. Yeah, this is a belly full of food.
Full of food.
Any...
Yeah. And it has those nice storage cells, those little circles. Yeah. Very cool.
And then here's the mouthpiece, which is made of calcium carbonate. So incredibly strong and powerful.
Yeah, it's a little tube and it just ejects out. Really kind of freaky looking organisms, really. The closer you look, the less cute they become, but isn't that true for everything?
Oh, and I was reading a paper about the mouthpieces of tardigrades, and there's, a tardigrade for every job, and there's a tardigrade that eats other tardigrades. So it has a particularly wide and capacious mouth because it has to swallow a whole tardigrade.
Yeah, and some they'll actually come over and they'll just kind of latch on and they'll just kind of pierce the skin of the tardigrade and kind of suck out the juices, which is something that's posted. And that kind of had mixed reviews. Some people are really interested by it. Some were just horrified and they just didn't want me to load that kind of stuff. So, yeah.
That's kind of brutal, isn't it? Yeah.
Yeah.
Yeah. And I posted a video of a tardigrade, dying, because as you see on the slide, the cover slip can flatten anything. So I had a tardigrade like in that image there, just kind of flat, and eventually the slide crushes it, and the tardigrade kind of explodes, and all the cells come out of it. And it was slightly gruesome, I suppose, but it was just showing these things aren't indestructible. Everybody thinks tardigrades can't be killed, but really they're quite fragile microorganisms. They just happen to be resistant to a lot of things which kill humans. But you can squash one.
Mm. Absolutely.
Not that you should, but it can be done.
How did that go down on the channel?
Very poorly, yeah, very poorly.
Yeah.
The point was made nonetheless. They're not immortal. They're just, they're quite resilient. That was the point.
So, what do you reckon? These tardigrades, they got spilled out on that mission around the moon, right? So they reckon that there's tardigrades there now, kind of dehydrated. What do you think the chances of them being rehydrated
Or resurrected?
Yeah.
I mean, there's always a chance. I don't think there's probably much food for them on the moon, but when these tardigrades enter this kind of cryptobiotic state, they have to do it slowly. It's kind of like, you and I can't just be thrust outside in the winter. We have to get our jackets on. We have to prepare for it, similar to tardigrades. So they can't just go from being happy in a tube to then crash landing and being at, a freezing temperature with all this radiation. So it might have just killed them instantly, but it'd be interesting to check it out, right? Go sample the moon.
Because then you get to the idea that what if this piece of moon rock was smashed into by an asteroid, and then you spent these tardigrade, what are they called again? What's the name of the dried ton? Is it a ton?
Ton, yeah.
Yeah, and these dried tardigrade tons then spread across the planet, the universe, and are spread to other planets slowly over time. That would really be, a consideration for.
Maybe that's how they got to planet Earth, right?
Maybe they do come from space. Actually, I wanted to show you and share with you, this is my laptop background, which is an amazing image of a tardigrade.
Little nose, yeah.
And you can see its mouthpiece. Where am I going to point? Over here. Yeah.
Nice.
Used for piercing bacteria. And then I guess there's calcium carbonate pincers stick out of the mouthpiece into its prey.
Pierce cyanobacteria as well, or just they'll eat any of the juices in the algae, or they'll pierce the cell walls of these things, and they're really interesting. And those little claws are great because they can kind of hook on, and so like the video you showed that I took earlier, the tardigrade, it was kind of just crawling kind of slowly, but that's because it was on just a wet piece of glass. You put it on a cyanobacteria strand, and I posted this before, they're a lot more agile, a lot quicker, and that's kind of how they're more naturally moving around. So that's a great image, yeah.
I mean, obviously, sadly, this tardigrade will be dead because it will be coated in gold for this electron microscopy image.
It's a great way to die if you're going to do it, right? If you coated in gold and immortalized in images. Yeah.
The thing that amazes me are these claws, these incredibly sharp, bear-like claws here.
Yeah.
It's got four. Yeah, I mean, they just look so mammalian. It's such a weird.
Yeah, it's crazy. And that one's probably maybe 0.5 millimeter or less in size. I mean, the average tardigrade is not huge. I very rarely see them above 800 or 900 microns. So yeah, they're really tiny little creatures. I actually have a couple of videos of some of these ciliates I worked on, which are 1.2 millimeters in size, swimming around the tardigrades, and tardigrades are smaller. So it's kind of neat to see this single cell, which is larger than this really complex creature with all these limbs.
Because parameciums, which I do have a video of here, are also quite large celled microorganisms. I think this one.
Yeah, maybe between 100 and 300 microns for a paramecium.
Yeah, I move over this side perhaps. Then I think in this next section there's a few bits of video.
All right.
Yeah, so here we go. There's paramecium. I think the next one you see some mating conjugation. So this is this is the original drawing of paramecium from a German scientific paper. And yeah, they're very complicated cells. They seem to just have.
They are, vacuole, little feeder canals. Yeah, brilliant.
Oh, I think so. These ciliates you're working with are very, very big, and now I'm learning that these water bears are actually a lot more aggressive and kind of gruesome than I realized. So, what would win in a fight between a water bear and one of your ciliates then?
Well, I've never seen that fight, but I think the tardigrade would, if it were to be ingested by one of these large cells, I think it would be able to claw its way out.
Oh, really?
Yeah, I think so, because it's kind of a slow-acting process, the digestion of these ciliates. I have seen some of these cells, a species called Berseria, which is basically like a big mouth that's shaped like a pumpkin, and that just goes around filter feeding, kind of like the rotifer, and it ingests everything. And I've seen that Berseria eat rotifers before, and these, rotifers, they have an eye spot, they're living creatures, and they just stuck inside the cell, and that's where it dies. So quite interesting, yeah. But I think the tardigrade would. But if it was small enough tardigrade, yeah, I'll look for that. Look to make one.
Yeah, cool. So I was thinking before when you're talking about how rare they are and sampling them. Do you have to do anything special when you're sampling? Do you have to do a lot of filtering or kind of have nets or anything like that? How does that work?
No, so I just take natural environmental samples, and then that kind of the sediments form in the bottle, and I don't shake them up or anything. I just keep it still, and I'll sample that water sediment interface layer. That's where a lot of the cells I work on like to be, in that kind of depleted zone. And that's where a lot of the tardigrades will be as well, feeding on the microbial mats. So I'll just take a little bit off the microbial mat, and from the right site, they will be in there in large enough numbers. Yeah.
One of my goals then is definitely to capture a tardigrade under a microscope. So I'll go looking for some moss when I get home.
Right. Or if you said you're in Europe, so in Northern Europe, if you find any of these kind of woodland ponds which have these microbial mats, so it's like this layer of either green or blue, just like filaments on the bottom, that would be a nice spot for a tardigrade to be feeding on. So I would take a bit of that water and take a look too.
Okay, I will search hard and far for these tardigrades myself.
Right. Good quest to have.
Indeed. So one other thing I wanted to ask you just before we run out of time is you now you have 140,000 followers. Have you ever found yourself recognized in public by people who follow your account?
Well, I was sampling with another Dr. Sally Waring at a microbiology conference in Canada, and we were doing some videos for Instagram, and the people there, they recognized us, they followed us. That was kind of fun, but that's a very specialized audience, people who are present at a microbiology conference. I've given lectures before to larger audiences, at ASM, that kind of thing. And when I start talking, they have no idea who I am, and I'll put up a slide of, here's my Instagram, and I'll see kind of in the audience, they recognize, and they might follow me, and that's kind of fun. But yeah, I think also it's nice that a lot of my account doesn't have many posts about me. It's more about the science. It's more about just showing off these cool creatures. Yeah. It doesn't really matter who I am so much. It's just, here's something interesting. You guys should all, take a look at this and hopefully respect the planet and the world a bit more, knowing there's all these creatures everywhere. So that's kind of been the fun part of it.
Well, that's a good experience. So I sometimes find scientists can be very scared that, if they put too much online and they become in any way internet famous, it will ruin their anonymity and their sort of like chilled out life. But it seems that you can have hundreds of thousands of followers and also live an ordinary life as a scientist.
Yeah, and if you recognize the scientists, what's the worst that can happen? They're going to
stop.
Can you see me?
I can. I'm just going to jump out from the microscope for the meantime. Okay, cool. I think we should pull this to a close because we've got a big group session after this with several speakers, and I imagine that they will try and go over time. So in order to keep them to time, we'll give them a few extra minutes.
Can you tell how many people are watching right now? Any idea?
I don't know. It's on several places. I think at the moment it's not huge. We've probably got, let's have a look. Where are we on? So on one of the YouTubes, we've got, just about 23.
Frozen again.
On a few others, we've got 5 here. This one's interrupted. It should resume shortly. So I can't see how many people are on this one.
Hopefully you guys can still hear me. A little bit frozen.
We can hear you now, yeah.
I reckon we've got 50 simultaneous viewers, and then we'll see at the end, because all of these talks can be rewatched, and then we're going to put them all online. But I could have a look back and see the individual peaks. So where did we start? We started around 4. Yeah, so I'm not looking at all the stats, but on one of the YouTubes, we've got between about 20 and 30 simultaneous viewers. And then on the other ones, I'll have to add them up at the end of today. But we're in about 6 places at once. Oh, I think I'm muted.
Oh, I can hear you.
Cool, there we go. No, I meant Hunter was muted. But yeah, let's move on to the next session because they're going to take their time. But it's been fantastic having you, and thank you for showing us your videos and helping us to identify things under the microscope. It's been great, and I hope that any scientists who are thinking about communicating with Instagram and microscopy have learnt a bit and can do it themselves from home.
Definitely do it. Why not? Especially in today's times. Yeah. Best of luck with your tardigrade search. Looking forward to hearing about it.
Thank you very much. And yeah, hopefully you'll get back in the lab soon.